Hey, it's Mr. YS, and this is a clip on DNA transcription. So let's have a look at this picture here, because this shows the end result of transcription. See if you can work out what this is and what this is. So what's happening as a result of transcription? Well, the top picture here, this is DNA in our double helix, and we're taking a section of that DNA, a gene, and using transcription, the whole process is, in the end, we're going to get a protein. So transcription is the first step in getting protein using the genetic code. We've also got another step, so the second step that we're not going to talk about today is translation. So there's two steps. Step one is transcription, and then step two is translation. Transcription uses genes which are present in DNA. So a gene is a section of genetic information which is running along, and that genetic information will allow the production of a protein. So it's not really a thing that we can zoom in and see it. We have to know what the code is to know, is it a gene? And there are the regions in between which are not genes. So there are sections which are genes and sections which are not genes. And transcription is using genes. And most of these make proteins. The other thing that can be made using genes are other things that we'll come to later, like tRNA, and also some of the components of ribosomes are made using genes, and those aren't proteins. But most of the time when we talk about transcription, we're talking about using a gene to create a protein at the end. If we zoom in on a region of DNA which we can consider to be a gene, which would be this region here, we can see that a gene is made up of a promoter region at the start, a terminator region at the end, and then in the middle of that would be what's considered the coding sequence. And so the coding sequence that's the information for protein, or those other things that we talked about, like tRNA or the RNA that helps to make up ribosomes. So a gene is this section, and at the start of every gene there are promoters, but the promoter and the terminator, these don't code for amino acids, and it's amino acids which would make up protein. So they don't code for that. And what they are, they're a kind of, the promoter says start, it says start here. It gives the enzymes involved the information to start at that point. It's a recognition site. And the terminator is the part that says stop here. So we've got two signals, the start and the stop. The start signal is the promoter, and the stop signal is the terminator. So in summary about a promoter, it's a non-coding sequence of DNA because it doesn't lead to the production of protein, but yet it's an essential sequence. And as we see next, the enzyme, it allows RNA polymerase to bind. So this is the RNA making enzyme. that's going to carry out transcription. And so this signal allows it to come and bind at the start so that it can then run along and it can create the RNA, which we call mRNA, because it's going to make messenger RNA, which is ribonucleic acid. So very similar to DNA, RNA is going to be made up of a phosphate. The sugar, instead of deoxyribose, is ribose. And then we have the nitrogenous base on it. So it's very similar. But we're going to see a difference in the nitrogenous bases because we're going to get a U for uracil. Okay, so promoters are not transcribed, but it plays an essential role in transcription. So if the question is, what do non-coding sequences do? Well, one of the answers is they are promoters and terminators. So they assist in regulating transcription. But what is transcription? What's this over here? This, if you kind of look carefully, this is a copy of grades for a student. This is a college transcript. So what's a transcript then? A transcript is a copy of something. It's a copy and it's in the same language. So here we've got a student's grade and it's copied. It gives someone a copy of the grades that they received. So if we remember that, transcription is the process of making a copy. It's not the original, but it's a copy of it. So the original, in this case, is the DNA. That has the original information. And the mRNA, that's the copy of the information. And just to slightly labour the analogy here, so here's a court transcript. So this is a court transcript. So a transcript, you can see here, it's just a copy of what was said. It's an exact copy of what was said. So same language, exact copy. So what we're going to do in transcription is take two strands of the DNA. We're going to use only one of them. There's only one strand which is used. And then as a result, we're going to make a single-stranded copy. So transcription is making a copy of the information from one strand. Here's our enzyme then, and in transcription we only really have to learn the one enzyme, which is lovely. We don't have all the enzymes of DNA replication. It's still called a polymerase in terms of making. It's at the same time, it's the builder, but it also has the same functions as helicase because it can unwind as well. So it's a builder and an unwinder in one. So we only need to remember one. For transcription, there's one enzyme that's really important, and that's RNA polymerase that does both the building, putting in new nucleotides, and the unwinding to separate the two strands. So in the first step of transcription, we get RNA polymerase recognizing this non-coding region of the gene. So it's still considered part of the gene. And it recognizes this non-coding region and it's going to come and bind. So we use the word bind to mean attach. So it's going to come and bind to this sequence. So although it doesn't quite show it on this picture, once it's attached to that sequence, we can say, yeah, RNA polymerase attaches to the sequence of DNA which is known as the promoter. So it's coming down, it's going to sit on that sequence, binding to it, and this is through attractions, interactions between amino acids in the RNA polymerase and the sequence in the DNA. This little bit will go a bit beyond IB, but for those of you who are kind of interested in thinking, well, how does it know? How does the RNA polymerase know where to bind? Within a gene, there is the promoter region. There's the region that we say is transcribed, so this is the coding region. And in the promoter are two recognition sequences which allow the polymerase to come down and bind. And those sequences are quite often, in quite a lot of genes, you can see they've called it the Tata box. So we get this repeated sequence, and this up here, TTG, ACG, we get these two sequences which are there at the start of a lot of genes. And those sequences, that's what it recognizes, and it goes and binds there before it starts. Once it's bound at the promoter, it will move to the point which is just past the promoter. So if this was the promoter at the start, it moves along just past the promoter into this region. And this is the region of the gene which has got the information to make, in this case, well, we're going to talk about protein. So what's it doing here? It separated the strands. So the RNA polymerase separates the two strands of DNA. And how's it doing this? So a bit more detail, it's the same as helicase. So helicase was separating DNA strands. So to do that, down the middle, joining these, were those weak hydrogen bonds. So it breaks hydrogen bonds. So it's not helicase in transcription, it's only RNA polymerase, but it's got the same function. So it breaks RNA polymerase, separates the two strands of DNA, exposing the DNA bases for pairing with RNA nucleotides. So floating around will be those nucleotides of RNA. And there are lots of these around, and we are in the nucleus, so lots of these are produced. So there's a high concentration of RNA nucleotides just floating around. There's also a high concentration of DNA nucleotides, but they aren't going to be used for this because we're going to make mRNA. An RNA polymerase then moves along, and in the kind of active site of the enzyme, it's able to allow these RNA nucleotides, the right nucleotide, to come in, and this is all enzyme kinetics, so it's just randomly colliding, and match the correct base. So it's going to start matching the bases, and it's only using this strand. So you can see the top strand is not being used. It's opened up the double helix, and it uses one strand, and now it's starting to put in a single line of RNA nucleotides. And just like with DNA synthesis, it's going to do this according to complementary base pairing. So we've got complementary base pairing where we have a thymine, if this is the DNA. The only RNA nucleotide which can come in and make the correct hydrogen bonds would be the adenine, the nucleotide with the adenine base. And so the only change that we get is here. So we see now, normally we would get in DNA, we get A to T with two bonds. But now we don't get that, so we don't have A to T in mRNA. Instead, we have A to U. So wherever you see a T, you replace it with a U instead. There is no thymine in mRNA. So if you were to see two sequences and you see one is A, T, G, C, and they say, what is this? Well, you know it's DNA. But if you see A, U, U, G, C, C, what is it? You know it must be some type of RNA, and probably messenger RNA is a good guess. So according to complementary base pairing, it starts to add in RNA nucleotides. And what's creating that bond in the middle? It's the same as in DNA. So between the two bases at the middle, so I'm just going to draw the one nucleotide, but between those two bases, what we have is, so this is the base. Between the two bases, what kind of bonding allows that? We've got, it's hydrogen bonding that's allowing those two, the DNA and the RNA, to make a bond. And it's only going to be a temporary bond. So they hydrogen bond together. So here's our summary of that. RNA nucleotides are matched to their complementary bases. So adenine now is paired with uracil because it replaces thymine. So we would get an adenine to uracil bond. The other ones would be the same, cytosine, and it's 3 bonds to guanine. And it's the hydrogen bonds between the RNA nucleotide and the complementary base on the DNA strand that hold them together. And this is something that you could, if you're answering this question, you could draw diagrams to show this and label the hydrogen bonds. Now, something which is kind of a high level, maybe 7 statement, is that we actually use nucleoside triphosphates. Nucleoside triphosphates. So if we think about what a nucleotide is, it's this. It's one phosphate, one sugar, and one base. But we're saying that we use nucleoside triphosphates. That's 3. So 1, 2, 3. So this is a very high level statement, but basically what happens is by hydrolysis of the 2 phosphate molecules there, they come off. So they then are released as those 2 phosphates. And through them kind of combining with water, we make more bonds, and that provides energy. So the energy for the reaction, for it to keep happening, is provided by hydrolysis of the 2 phosphate molecules from nucleoside triphosphates. Now, if that's too much, you don't really need it because that's a very high level 7 statement. If you had to guess, what direction would you guess that new RNA nucleotides are added? If you know about DNA replication, what would be the direction of addition, of adding RNA nucleotides? It's going to be the same. So if this bottom strand of the DNA is running 3' to 5', then the top strand will be running 5', and the end here where we're going to add the new nucleotides is going to be the 3' end, just like in DNA replication. So we add in new, RNA nucleotides at the 3' end. Nucleotides are added in a 5' to 3' direction. And then the polymerase will just keep running along the gene, adding in. So as it's doing this, we don't show any of this, but these nucleotides, these RNA nucleotides are just, there's loads of them and they're just happening to collide with the active site at the right time. They're all flying in and being combined, and then we've got the mRNA underneath, which is being created. So it's just going to keep running along until it needs to stop. And the place where it's going to stop is this, the region that we talked about, the other non-coding region, the terminator. So when it gets to the terminator, you can see the polymerase has detached, or has been released. Or the other word, the big word, dissociated. Maybe correctly spelled without the extra S. So dissociated. So the mRNA has also been released. So at the terminator, the RNA polymerase gets released, the mRNA gets released, because this, the mRNA, that's our transcript. That's our copy of the DNA. Now, why is it a copy? So how does that relate to what we saw before to do with the language? Well, it's the same language. What language is the DNA in? The DNA is in, it's the language of nucleic acids. Deoxyribose nucleic acids. So the language is nucleic acids, and the copy, the RNA, is in the same language. Apart from uracil, which replaces thymine, it's exactly the same language. So it's a copy. It's a single-stranded copy of one of the strands in the DNA. In summary then, at the end, the terminator is a sequence of DNA signaling the end of transcription. The RNA molecule, and its mRNA, completely separates from the DNA. And we also saw that the RNA polymerase is released as well. Now, anybody wondering how does it know to detach? What happens is, in some cases, a loop is created. At the end of the gene, in the region of the terminator, there are lots of G's which are transcribed, and C's, and these G and C's come back together and bond together. So if you've got like a G here, you've got a C over there, G here, a C over there. They create a loop, and this loop kind of makes it fall off. But that's beyond IB, but it's basically a sequence that creates a loop that causes it to fall off. That's one way. And so at the end of transcription, we have an mRNA molecule, a single strand of RNA nucleotides. This is our mRNA molecule. But the question is, well, what did it copy? What did it transcribe? Because it transcribed one strand of the DNA. And so how does that work? If DNA is double-stranded, why did we only use one strand to get a single strand, the single-stranded mRNA? Let's have a look at these two sequences. So let's imagine these are the two strands of DNA. And if we've opened them up, have a look and look at which one actually has information that we could read. So I've deliberately chosen some information that we could read. One strand has information, usable information. Which strand is it? It's the top one. So we've got cat, tag, cat. It makes some sense, we can read it. But the other side, GTA, ATC, GTA, that doesn't make any sense. So there's one strand which makes sense. It's got some kind of language in there. And this we call the sense strand. Now, rather than being like English language words, if these words really had the sense to give the information for an amino acid, and that's what it is, it's a triplet code. 3 bases can be read as one amino acid, one of the 20 amino acids that it's possible to have. Whereas if we read this, it doesn't make sense. So we can call it the anti-sense strand. So we call the top strand the coding, because it has information. So when it's got information, we call it coding. The coding or the sense strand. And this bottom strand is the anti-sense strand. Okay, but then if you're RNA polymerase, which strand do you use to add in new nucleotides if you want to get a copy of the strand that makes sense? We want to get a copy of something that makes sense. We want cat, tag, cat. So which strand do we need to use to make mRNA? Where are we going to put the new strand via complementary base pairing, our new single strand, to get a copy of cat, tag, cat? If RNA polymerase was going to come and bind and then use the top strand, if it used the strand that makes sense and put in the nucleotides, it would get the message GTA, ATC, GTA, and that would be the mRNA, and that would go off. But this doesn't make sense. So if we use the strand which has the sense on it to make the mRNA, we get something that doesn't make sense. You can imagine this like a little bit like a mirror image thing, because complementary base pairing, we're kind of getting like an opposite. We're getting the mirror image of it. It's not quite like that because we're just getting something that fits with it, something in a pair. So we can't do that. We can't use the coding or sense strand to make the RNA. So instead, we use the antisense, which a nicer way to maybe think of it is the template. It's the template that we use to make RNA which has the same information as the sense strand. The sense strand had something that made sense. It had cat, tag, cat. So if we want to get that back, we need to use the one which is the opposite to it. We need to use the template strand. So here, if we then go and add in, we get C pairing with an A. Now the only complication here is that now we're going to change, we don't get T's. So it's going to change the message slightly, but we're still following the language. And by pairing again, we're going to get U and A and G. We're going to get C and A and U. And so what we're getting is the mRNA copy of the sense strand. So this mRNA that's going to go off now has the same information. Now, unfortunately, there's no good examples where we're not changing the words, because it would be great if it came out as exactly the same, but we've changed this at the end. So we've got two changes there, and we've also got our change here because of, so yeah, there's 3 changes. So it doesn't say exactly the same, but in mRNA we can say that a U says the same thing as a T. So we've conserved the sequence. And so transcription overall is the synthesis of an RNA molecule that's identical to the coding strand of the DNA, aka the sense strand. And to do that, we need to use the one which is the opposite. The antisense strand acts as the template. So here's our RNA polymerase going down and using the template strand, or the antisense, to then get a copy which is the same as the other strand, the coding strand, which actually has the information for amino acids. So let's just have a look at this example here. If we follow it through, if this is the strand which actually has the information we want, the coding strand, then we use the opposite one, the template strand, and then you can see that the mRNA that we're going to make has the same code as the coding strand all the way along. It's going to be exactly the same, but we change any T's which were there, we're going to change for U's. So all we have to do, if we're thinking about what will the mRNA be, and we know which the coding strand is, then we just look along, and any time we have a T, we change it for a U. And that will then be the sequence on the mRNA. So the mRNA equals the coding strand, or the sense strand, with U instead of T. And if we look at it as a whole, and we'll get onto translation next, it means we take the DNA, we open it up, we use the template strand to make our RNA, which has the same sequence as the coding strand, but with those U's in place. And then that can be read off in 3s by our ribosome in our next step that we'll have a look at. And it can read 3 at a time, and each 3 letters is going to tell it which amino acid to put in. And therefore, if we know which amino acid, we can build up a long chain to make a polypeptide, which can fold into a protein, and then it will have that specific function.